Okay, shall we get started? So last session of the day, which is high throughput crystallography for ligand screening. So topic that's very close to my heart. I should mention we have a change to the program. So the fourth talk, which was going to be by Nick Pierce, is not happening, but we have got a substitute, and that's Stephanie from the organising committee, so she'll be stepping in to give that fourth talk. Otherwise, I'd like to introduce Ailsa Powell. So Ailsa is a principal industrial liaison scientist from Diamond Light Source, and she's going to talk to us about fragment screening at the XChem facility with industry. Thank you, Ailsa.
So first of all, I'd like to thank the organising committee for inviting me here to talk about this. It's unusual for the industry team to get let out of the cupboard and talk at meetings. So it's going to be a little bit of a different talk to maybe the ones you've seen earlier today. There's not as much data. In fact, there's no data. So sort of when we're working with the commercial projects and a lot of users coming in, we sort of bound by the confidentiality. So what I'm going to talk about is first of all an overview of crystallographic fragment screening and the XChem platform at Diamond. I'm aware there's a lot of young scientists here who may not be so familiar with what we can offer at Diamond, the concept of what we do with crystallographic fragment screening. We saw some excellent talk from Rob yesterday, which was like a little showpiece, thank you. And then the discussion following afterwards is like a really nice way of sort of thinking about how all the different techniques we can do for high throughput screening in early stage drug discovery can kind of come together to sort of feed towards that sort of lead optimization that we're looking at. So, and the second part is kind of alluding to what the organisers asked, is how the industrial use has changed of the XChem facility. Now, I can see a few of my users in the audience, and there's some of the academic users as well, and we'll just kind of sort of be able to sort of gauge how it's adapted and actually how it's quite different from the academic programme, if that's what you're used to. So, starting off, get a laser pointer up, sort of kind of want to sort of talk about what we're sort of talking about when we're looking at structures. We've seen some really good talks on this so far in the meeting. So I kind of just want to highlight a few little things. And this is just some data pulled from this sort of selection of papers down here, which are kind of they're reviewing the published sort of products of drug discovery programs that's coming through. Now, sort of in a yearly review, we've got one from sort of 2022, and then in December last year, so 25, the stats came out for 2023, and I thought this was quite an interesting comparison. We're here doing X-ray stuff on the whole, this meeting. It's a little bit disturbing, this drop that we see here in the fragment hit identification for X-ray based stuff, especially if you're me. But I'm a bit of an optimist and I'm not too worried about this. What I found really interesting was this surge in NMR. It's like we're zooming back in time, and the NMR screening, which is a very powerful technique if you're looking at sort of ligands and fragments, has been really popular in this. We're also seeing mass spec screening pop up for the first time on this, and sort of various changes. Now it's a small but I use this as an introduction and the end, maybe, of my talk. Okay, help is on its way. So just to sort of show that X-ray fragment screening is one of many. It's part of a cog in the whole process, sitting in and getting that early chemical data ready to move forward. And we're back. Maybe don't go too far away. So, for anybody who's not familiar with what we're talking about with crystallographic fragment screening, we like to use the idea of Lego bricks at Diamond when we're explaining this to people. So you're talking about small units that you can kind of look at how they fit into shapes and then build it together. So to get my pointer back, maybe. Okay. I can see what I'm doing, but you guys can't. Right, I'll just carry on. So with the idea that you get this data, you can look at it. And yesterday, I think you're probably getting a feel for how much data we can be talking about here when we're looking at structures. But once you can filter it through, you can start looking at what you have. You can try sort of growing your fragments, linking them together because you've identified these different binding pockets. And then if you've got kind of really nice fragment sets, you can try merging them together. It's basically generating information for your chemists to kind of like push forward, and feeding the chemists is what we're kind of doing in this business. So it's sensitive. You're generating structures as well as sort of the binding information, and that can really help drive projects forward in conjunction with some of the other techniques. And again, I'm going to reference you back to Rob's talk yesterday because I think it showed some really nice examples. I would like to point out that fragments are weak binders, and we really hammer them into the crystals. So with the XChem facility at Diamond, this is a very kind of sort of broad overview. You have a library plate of fragments, which you can see at the top left there. You have crystals, a lot of them. You combine these in a soaking scenario, which we work out, and then we actually manually mount them at Diamond using shifters. So I'll explain a little bit more about what they are. Then we move them across to the beamline where they're collected on automated data queues. There's an arrow explaining an awful lot of data analysis here. And then your output, here we have a very kind of like overachieving active site groove with a lot of fragments in it. Sort of moving forward onto a more detailed view of what the pipeline is and what we actually do within the lab. We have XChem ready crystals. Now we have some fairly high standards when we're looking at this. So we're sort of looking at, you need thousands, at least 1,000 to start with, sort of relatively same diffracting crystals. So you're looking at nice diffraction, small range, basically identical. So you can get really nice comparisons in your data analysis further down the line. We have a fragment library. Now, these go in at high concentrations. So our fragment, so a basic library set is at 500 millimolar. So we can be going in at quite high, we can be looking at sometimes 150 millimolar going into your crystals, which is, again, reflecting that weak binding that we were talking about. Then we enter the platform. So we image the crystals, target the crystals. We dispense the compounds using an echo dispenser, which is an acoustic dispenser sort of firing up to our targets within the drops. We harvest the crystals using the shifter, which is an automated platform with integrated software, which means we don't have to make notes as we go through. We can get up to 200 crystals an hour using this on a nice crystal system. Talk about perfect crystals here. Less perfect, really about 100 an hour, which is still pretty quick. Then we move across to unattended data collection on our beamlines. Then that diffraction data enters into the data processing pipelines, which again, all automated at Diamond. And then the box here that says hit identification, that's all the data analysis. So you're looking at taking that data, curating that data, processing it through Dimple. We use Panda as our analysis tool for identifying hits, then fitting the hits, so those ligands that are bound in, and then running refinement. And then once you've done all of that, oh, I've got a pointer back. it's not moving. Sort of then we disseminate the model, and who we're disseminating that to depends. So if you're an academic group, you might be going for a PDB deposition for publication. Industry will be taking that data in and putting it through their analysis pipelines. And kind of we work in different ways for different users as well, which will hopefully come back to. So for those of you who have not seen our lab, we have it here. We have a couple of echoes in the back where we do the compound dispensing. We have 4 shifters in total. You kind of see them sort of sitting around here, and then you can see the plates going in and the sort of harvesting process. We have compound storage, so we can store compounds under low humidity and oxygen environments. And at the moment, we have capacity for 4 users simultaneously. So at the moment, that's in-house development for 1, 2 academic users, and 1 industry a week. Then we move across to our beamlines. So here at the bottom, you can see exactly how close our beamlines are to the lab. We have Izzy from our group here wheeling our storage duo across from the lab, which is just behind her, into IO41. So it's literally across the corridor. You can't really get much closer. Any of our beamlines that are UDC enabled, so Neil on IO3 takes some of our samples as well, can run the XChem queues. It can have 36 pucks per load. Multiple projects can go in onto a load, and we run the automated queues. And for anybody who's not aware, IO41 exclusively runs the XChem queues, so we don't run generally any other data collection on IO41. Now we've gone over the overall workflow and the pipeline, but we run that multiple times per project. So we start off understanding the solvent. So generally the compounds we have are dissolved in DMSO, and we need to understand how the crystals can tolerate the DMSO. So we do a set, usually we'll need about 50 crystals for this if there's cryo involved as well, running 5 to 30% DMSO, and then a couple of time points. And then we will do data diffraction tests on that, so we can see how the crystals have tolerated it, if they're dissolving, where the cutoff point is, basically how hard we can push those compounds into the crystals. So providing we kind of get sort of keep our crystal integrity and still got good diffraction coming through, we can move forward onto a pre-screen where we take a subset of our libraries, run those through and check that we're still getting diffraction at the end. When you start adding the compounds in as well as the DMSO, you can start affecting your crystals, and occasionally they can dissolve, you can lose your diffraction quality. And from that point of view, we will then go back and kind of like ease off a little bit on our kind of potentially quite harsh soaking conditions and kind of go in a little bit sort of gentler. Again, ideally we will analyze that subset, and we should be able to see some hits coming through from that when we analyze it. If that's all good, then we can go ahead to the full screen, and that is like as many compounds as we need to put into that. Again, once we've collected all that full screen, we go through data analysis, and you can see a readout here for the MPRO protein from SARS-CoV-2, where you can see across the surface, and I think you'd see this in Natalie's talk a little bit earlier, where all the fragments can sit in as well. So you have a main site, and then they fit into pockets all over the surface. So you can't say, I'm only looking, you can say you're only looking at one site, but you need to sort of bear in mind that the fragments go all over the place. Generally speaking, you might get a 5 to 10 hit rate off your fragments on your protein, but it depends on the project, how you've curated those fragments going in. That can have quite a big impact. And then you, like I said, you'll get them at a range of sites. So how have women doing? XChem has been running now for 10 years. So we're looking at sort of 300 academic projects, a ridiculous number of data sets, a lot of liganded structures, a lot of PDB depositions. I would say that some of these, so if you look at the PDB deposition graph at the bottom, As a result of the in-house research we've been doing on the antiviral work in ASAP, this has really been pushing it up in the last year as the structures for that get deposited. We've also completed around 200 industrial campaigns in this time. And if you want to sort of check out some of the stuff we've done recently in a recent paper, there's one there at the bottom. So then looking at our access routes very quickly, we have a standard academic route. I think some of this was covered yesterday, but just to make it clear that we sort of run 3 access routes, standard academic, the bag access, and then the proprietary access. So standard academic and bag go through the peer review process, proprietary access, you contact me. And we support in-house research as well. So now I want to move on to this evolution of the industrial user experience on the XChem platform. And it's going to take a little journey back in time and then through to the present. So in the beginning, the completely parallel industry program started in 2018 with an expansion of the lab and recruitment of staff, which was me. And then it sort of modeled the academic approach at the time. We had users coming into the lab. We had 1 user a week because of that, and had 2 beamline shifts, which only at the time allowed us to do about 300 samples a week. There were drawbacks on this. Significant time pressure on getting that solvent test, understanding the DMSO effect on our crystals, which we needed to do before doing the main compound soaks. And then we sort of very quickly resolved that by moving that component, breaking it free from the rest of the screening and doing it an earlier session, even if that meant the staff were running that test rather than the users. There was often not enough time to squeeze in that pre-screen, which is actually a really valuable tool in understanding how the compounds and your crystals behave together. It's not unusual to tweak your soaking conditions in that step. It also allows you to gauge how successful your campaign, your project might be. So you could know that you're going to be looking at hundreds of fragments going in, or maybe a small percentage like 10 or something. Often we didn't have enough time to analyze that, even if we could get it done before having to move forward to the full screen. So there were limiting factors here. One of them is obviously the beam time. 300 samples a week is not even our full library at the time. Lab availability, to do those due diligence on the pre-screen, sort of crystal readiness on a highly scheduled user program. And what I mean by that is your crystals have to be ready on a set date in 3 months' time. And we need a lot of them ready then. That's quite a demand. And if it gets to the point the week before, you're like, we lose the beam time, especially if people are doing larger screens. That could be a couple of weeks of beam time we can't fill. And because of the way we were working, we had a 6 to 9 month waiting list, which is a huge problem if you're looking at feeding into sort of an industrial drug discovery cycle. So how did we evolve this? And then sort of change where we were going. So there were new beamline upgrades, new detector, we had a new insertion device, and that meant faster data collection. We also had everyone fully move on to XChem queues. So there were no more scheduled visits, so no more of those shifts, kind of, and a dedicated, you've got 16 hours this week and that's it. And we had beam time access whenever we needed it. So it means we can, if our samples are ready on a Thursday, not a Wednesday, we can actually run them rather than wait another week. So we also moved to a full service model. So before we had the users coming in, this was a consequence of COVID. It was very much a switch. So we couldn't have users coming into the lab. And whereas we had been gradually moving in that direction, COVID was really the lever that sort of pulled it into this first full service mode. But what it meant is we could stagger the projects. So it was about the project management. We could have many projects in the platform at the same time because the staff were running them. We didn't have users coming on site. And it meant we could get through a lot more projects. It also slightly changed the way we could function, and it made us more efficient. But the instant result was an increase in throughput and no more waiting list. You can come straight in and we will start working on your project. So sort of summarize what this has achieved, the limiting factors I mentioned earlier, beam time, not a problem anymore. That 6 to 9 month waiting list, not a problem anymore. Crystal readiness and highly scheduled program, not a problem anymore, but crystal readiness is an ongoing thing where you have to have your crystals ready. But what we can do is move around that. So if a crystal system isn't ready when we were planning to do that experiment, we just move another project in, work on the crystal system with our users, and then kind of keep moving forward. So it makes it a lot more efficient. And kind of what does that give us? It gives us flexibility. We can move very quickly. And I will apologize some ways for this amusing and disturbing AI picture of protein doing yoga. So in case anybody hadn't quite picked up what that was. But what this has meant is we're more aligned with what industry users need from high throughput crystallographic screening. It means that we can move at the rate that that data is still relevant. However, I'd say what it's done is it's shifted bottlenecks. So what we started off with is beam time was really the bottleneck we had there. Then we eliminated that problem. Then we had lab capacity. It's like, how can we get these projects through with our staff? We got more staff, and that eased that problem. Now, our current problem is still the crystal systems. It's an ongoing thing, and then sort of looking at how you can really get those robust systems, because you will get better data the better the crystal's going in. So it kind of like how we can work on that, and it's a general problem, not just for industry, it's for the academic users as well, but it increases your success if you get that crystal system right, and it will determine how you can get there. So I'm kind of going to zoom through this because my 5-minute warning's up. So how has this changed how we approach sort of how the change has changed how it's industrial crystallographic screening is done at Diamond? So what about the project and the targets? So it depends on the nature of the company. So we work with all companies of all sizes, doing all sorts of things. So you have some coming in on a selected project. It's highly sort of triaged, very carefully selected for suitability with potentially a specific question about that target. Whole portfolios. Once people have realized how quickly we can turn out data, they've dumped everything on us. And we churn through it, give them all their data, and then don't hear from them for a couple of years. And then we get another load coming through. As an integral part of a screening pipeline. Now, Rob yesterday demonstrated how you've got the biophysical, biochemical, virtual screening, but you can also throw in your X-ray screening. And what we're seeing is people doing all this in parallel now. So they've got all the data in one go, rather than maybe doing your biophysical, pulling out some data and pulling through that way. So I was delighted to see Rob's talk yesterday because you provided me with some lovely examples. And then the last resort. These are the heartwarming ones where there's been nothing from any assay anywhere, no chemical matter. The people have come to us and we have got them hits. We have got them something to work on and save the project. So those are great. Now, why would you do this? So again, depends on the nature of the company. You've got the new target, new information approach. You've got the hunting for new scaffolds, old target, new information. You've got alternative binding sites, old target, new information. And then you've got structural information on the compounds that you've identified from other methods, your other screening methods, which isn't so much the sort of typical what you might think of as screening, but you're still using the platform we have to generate large amounts of data. And then the screening around known compounds. So you've got something bound, but you want to probe all the other sites. So you can put in your compound, fragment screen around it. Similar idea to the back soaking that was mentioned yesterday as well, but without back soaking. And then what to screen this follows into. So we have our DSIP in-house library, which sits about 850 compounds at the moment. We have users bringing in their own libraries. We have the other libraries in our portfolio, and you can see in the website I've listed at the bottom, all our fragment libraries. You can do compounds from other screening methods, and we also take follow-up compounds. Now, you can do all of those, mixtures of those, any which way you can think of, you can juggle that about, you can bring that in, and we can generate the structural data for you. Now, people sometimes don't think about doing their follow-up screening with us, but I just sort of want to kind of emphasize the power of what you can achieve by doing this. We can take large compound sets. So if anything from, we have taken 20, which is small, up to 600. So you can really start looking at generating data on your chemistry that you've got there. The crystal system's already established, makes it nice and easy to bring in-house and sort of, if you've done your screening with us. The diffraction data is ready very quickly. And the success readout is available within days of receiving those compounds in the best of scenarios. And we can be adaptable as the projects change and the compounds evolve through time. So the key benefit is that it's rapid feedback for the chemistry process, and we can kind of cycle through things really quickly and generate, in some cases, some amazing results for our users. So just to sort of summarize what we've got here, because I talked about efficiency and impact, and I hope you've been able to see the efficiency and sort of the implied impact that we've got here. So we've evolved our user program, and it is different from the academic program. It's been driven by the innovations at Diamond, how we access the platform, or how you access the platform, and how the projects are managed. Our efficiency is the dedicated beamline and lab and team. The adaptability we can do on a project by project basis. I don't think I've ever had 2 projects that are coming in basically at the same. And we can run multiple projects at one time. And we've removed our waiting time. So the onboarding, if you've got a crystal system ready, is pretty much immediate. And we can get that solvent readout done, know if we can move ahead. The impact is that rapid readout, very quick results with structures. It allows for very quick evaluation and very fast chemistry. And then the results are available while they're still relevant to the project, and not 6 months later when all the sort of SBR assays have already generated the chemistry and no one's interested in the structures anymore. So with that, I'd like to acknowledge, I'm going to refer back to Dave Hall's slide, I think, a little bit there, because he had everybody listed, and sort of summarize the sort of the industrial liaison group at Diamond, specifically the MX team members, past and present. Within that, Elizabeth for sort of championing sort of the development and how we could grow this. The XChem IFL1 team, again, all members past and present. Frank, who has sort of developed that project, came in, XChem was sort of created, and it's really grown, as you can see from the open bind sort of initiative that's coming forward now, and the rest of the Diamond MX group, who sort of allow all this data to kind of really process, plus all the backup teams in computing and everything like that.